Welcome, everyone. Thanks for joining us today in celebration of Darwin Day. You are here to attend the Celebrate Darwin Day with Biofuels and Mushroom Habitats, hosted by Vernier and Biorad. I'm here with our hosts, or our presenters, John Melville and Damon Ty, and I'll be introducing them a little bit in a few minutes here. So today's agenda, we're going to do a quick poll here in a moment. I'm going to tell you a little bit more about your presenters, and then they're going to introduce you to the Biofuels Enzyme Kit and how it can be used with the evolution of cellulose, I probably didn't say that right, activity. They're going to show you how you can use this activity for remote education, whether you're in the classroom or if you're in a hybrid or a fully remote setting. And then we'll be giving a demonstration of how you can tie in Graphical Analysis Pro. So I'm going to launch a quick poll that will ask you 3 questions. And if you could answer those. So we just wanted to get a little bit of background information from everyone to see if you are familiar with Biorad and Vernier and get a little bit of a better idea of what grade level you teach. So I see a lot of people are putting their answers in. We'll give it a little bit more time. It looks like most people are definitely familiar with both Biorad and Vernier, and it looks like we have a little bit higher percentage of high school educators attending, but with a good solid number for college as well. And I don't see any for middle school. So thank you everyone for participating. I'm going to end the poll now. And then I will introduce John Melville with Vernier. He has a BA in biology and psychology from Sonoma State University. He has a PhD in zoology from Oregon State and a postdoctorate fellow from University of Oregon. He's an associate professor of biology. He has major and non-major courses in biology and neuroscience and active learning with emphasis on data collection and inquiry. Damon Ty from Biorad will be the second presenter today. He has a BS in biology chemistry from St. Mary's College in California. He taught high school in Portland, Oregon, a genomics researcher at the Joint Genome Institute for 10 years, and 10 years at Biorad as a curriculum and training specialist. So I am going to pass this over to John and Damon for the presentation.
Great. So I just wanted to say that Damon and I have worked a lot on Biorad activities and also working on this activity. We just want to let you know is that we're here for you. We like to say that Vernier is for teachers and by teachers, and we love working with Biorad because they have the same sort of ethos. They're really here to help you. We've worked with them for a long time. If you can ask all of your questions in Q&A, we encourage you to do that, and there will be someone there that can help. If time allows, we will also discuss questions at the end of this talk. The other thing that I would like you to do, I'll show you how to do this later, but if you go to vernier.com and go to downloads, you can actually get a free copy of Graphical Analysis 5.4. You can download that to your device. There's no sensors required. We're going to be doing an active activity where I'm going to be sending you data remotely over the internet at the end of this talk. So go ahead and try to download that now. And then with that, I'm gonna let Damon begin to talk about this really cool activity, which is in our Investigating Biology Through Inquiry book. We co-developed an activity with Biorad that used their Biofuel Enzyme Activity Kit, and it uses our GoDirect SpectraVis Plus to look at the evolution and ecology of fungi. So Damon, go ahead and take it away and just let me know when you want me to advance slides.
You got it. So for those of you guys that are not familiar with Biorad, we have a lot of these education kits that basically come with everything you need in one box. And this is one very similar to that, where all the reagents you need, all of the plastics you need, all come in this one kit. And then, of course, we pair it up with Vernier's wonderful SpectraVis Plus in order to do more of the high-end analysis with this lab. In general, what this lab is going after understanding enzyme kinetics, but there's this 6 levels of activities that you can run with this. And one of those specifically looks at basically bioprospecting for enzymes that are used to break down cellulose in the production of biofuels from fungi. And the reason this fits really well within an ecology or an evolution class is that understanding the ecological niches of those fungi will usually dictate the type of kinetic structure you see from the cellular bias that you can extract for them. And so it's got this really kind of cool potential to do, you know, evo-eco type stuff, and then bring it all the way through down into the biochemistry by looking at these enzymes' kinetic rates. So it's a super cool lab. What I love about it too is that you've got at least 6 activities you can do in there. And so I do have some instructors that will use this for multiple weeks as they kind of go through looking at all of these different activities. And that's everything from looking at how does pH affect these enzymes, how does temperature, how do concentration, substrate, or the enzyme affect it. And so there's all these different directions you can take it. Today, though, we're just going to look at what traditionally was activity 6 in the first kind of version of this kit, and is actually the hallmark of this kit that we call the Biofuel Enzyme Kit, which is the bioprospecting portion of it. So go ahead and go to the next slide, John.
Hold on.
Yep.
Oh, there we go.
So this also is a kind of a cool step into an entire kingdom that we rarely talk about in science education, which is the kingdom fungi. Which produce a lot of these enzymes that in the next 10 to 20 years, we're going to see a lot more development as that organisms in this kingdom produce a lot of enzymes that you just don't find anywhere else that do very novel things that humans want to be able to do. One of those specifically is take giant amounts of biomass, that's mainly cellulose, right? And then break that down into useful molecules, i.e. if you can get it to sugar, you could basically turn it into anything you want, including a fuel. And so what students are going to do with this is they're going to go bioprospecting in the portions of the fungal kingdom that are easy for all of us to access, which are the basidiomycetes and the ascomycetes, which are the portions of the fungal kingdom that produce fruiting bodies, these things that we can actually walk around out of the forest and find, or from a ease of use in your classroom, you could just go to most grocery stores now and find a number of species there that work perfectly with this kit. So go ahead and go to the next one. So what I like about this is that it helps students also think about not only this kingdom, but think about this kingdom within an ecological context and an evolutionary history context. So if I were to walk out into a forest near me, this is a picture actually from the hills near where I live in Oakland, California, I can find fungi, these large, actually mushroom producing ones, in different little niches in the habitat. And the reason that these guys ended up here is through evolutionary histories that allow them to exploit getting resources in those specific locations. And so in an oak dominated forest like this, I could find kind of at least 4 different large macro fungi producing mushrooms, basically holding down very different ways of getting their food. So the first one on the top there that's all shaggy, that's called a lion's mane. That one is a parasitic mushroom. So it is actually using some of its energy to fight a plant while it's also eating the plant. If we go to the one beneath that, that might be one some of you guys have actually consumed. This is a chanterelle mushroom. This one's not beating up the plant to get the food. It's like, hey, let's work together. I'll kick you some, stuff that's hard for you to dig up, some of these micronutrients and stuff, and you'll kick me back some sugar. And so this one doesn't have some of those enzymes that might be necessary to actually break down the wood to get food because it's getting a handout, but well, it's actually getting an exchange, right, in the symbiosis. You can also find things like oyster mushrooms easily in every single habitat in North America, but definitely on oak trees. And these guys are there just eating a buffet of dead wood. So these are your classic sapotrophic fungi. They're there just for the cellulose buffet. They're not fighting the tree. They're not making deals with the tree. And then you've got other kind of decay mushrooms that aren't going after these really hard, woody substrates, but will go after stuff that's kind of already decayed a little bit. So the Agaricus mushrooms that we're all familiar with from the grocery store, those are growing on kind of what I like to call pre-processed cellulose substrates, i.e. poops and things like that. And so you've got in any habitat, you can find these macrofungi occupying these very different niches, and each one of those will have very different activity when it comes to looking at their cellulases, or specifically the cellulobiase enzyme that we'll evaluate in this lab activity. Go ahead and go to the next one. So most of you guys, because you're teaching biology, are very aware of what enzymes do, right? They basically decrease your activation energy. A lot of them will do this through a positional fit. Cellulobiase, the enzyme we will be looking at, does exactly this. It basically grabs cellulobiose, which is a dimer of glucose, beta-linked. It basically presents that in such a way that water can easily attack that bond, and you can now break that apart and have 2 molecules of glucose to work with. Go ahead and go to the next one. The United States has actually put a lot of effort into this over the years. So it happened with the first oil crisis at the end of the night or into the 70s. It happened again in the early 2000s. Basically, anytime oil prices spike, the United States dumps a bunch of money into R&D, trying to figure out how can we turn all this biomass that we're great at making into fuels. So that way we're not digging up dead stuff to burn. And it also then has a much better, basically, carbon signature too, because you're capturing the carbon that you then end up burning. And so the US, when I was working at the national labs, we worked a lot on trying to figure out how do we take biomass, like this tree that you can see in that picture, that's a poplar tree. So that was the first tree genome ever sequenced in 2008, exclusively for the desire to turn this thing into biofuels. A big portion of this kind of equation, though, is not only how do you find the right organism to grow for that, but really how do you figure out how to break this down cost and time effectively. And so there was tons of research going into how do you model this, how do you test this really quickly, because we're pretty good at breaking trees down. We've been doing that with the paper industry for years, but then taking that cellulose and breaking it down further into glucose so that you can then ferment that into any of these fuels that you might be interested in, is where a lot of the R&D is going on and has been going on for the last 10 or 15 years. And your students can kind of participate in that by looking at local fungi or fungi from the store and seeing how effective they are at converting that cellulose potentially into glucose. So go ahead and go to the next one. So if we get down to the kind of the nitty gritty of what this looks like. So if you've got biomass, if you were to kind of zoom really far in and look at the goods, really what you've got is you've got a bunch of crystalline cellulose, which is really hard to work with. Enzymes don't have an easy way of getting in there. And this is why you see logs in the forest. If you didn't have that crystalline cellulose, those logs would get processed really, really quickly. But what has to take place is that crystalline cellulose kind of has to be broken apart into a linear form for enzymes to kind of start getting access to it. So in industry, what we do is we heat up that crystalline cellulose in the presence of acids and all kinds of stuff to basically kind of denature it more or less. And then we can start dumping enzymes in at it. We use a whole suite of cellulases. So cellulase, basically ACE enzyme, cellulose at the beginning there. So these are all enzymes that break down cellulose in different ways. So some of these can basically trim it in from the ends. Some can come in and cut in certain branch points. But ultimately what you get down to is usually these dimers of cellulobiose, where there's actually not a huge number of enzymes, or from a niche point of view, there's not a huge number of organisms that produce enzymes that will break this 1,4-beta linkage down between these glucose molecules. And this is exactly the enzyme that your students are going to be looking at. So go ahead and go to the next one. So researchers, though, were having a hard time when they first wanted to look at how does cellulobiose break down, because it's always best if you've got a quick kind of field test you can do, right? That you can go, okay, I can see this being broken down. The bummer, though, about working with cellulobiose, so actually most of you guys have probably worked with glucose before, and you know that it more or less looks like this. It's more or less a clear liquid. Cellulobiose, unfortunately, is also sort of a clear liquid. So unless you've got your portable proton NMR machine or mass spec with you in the forest, it's kind of hard to easily diagnose when an organism has the enzyme to do this. And so one of the major kind of developments that came out were chromogenic ways that we could monitor this reaction. So if you go to the next slide, the substrate that the Bio-Rad kit's going to use is one of these ones that was getting used in industry to do this. So instead of using cellulobiose, we're using an analog to it that is basically glucose bound to p-nitrophenol And the beauty is these cellulobase enzymes are basically promiscuous enough that they will accept this as their substrate lover. They'll grab that, they'll cleave it, and you'll get a molecule of glucose and a molecule of p-nitrophenol. And any of you chemists out there that have worked with p-nitrophenol, you know that in a basic condition, it turns a nice bright kind of yellow color. So it allows you to do an easy colorimetric kind of measurement of how this reaction is progressing. And so that's exactly what we use for this lab. We're going to use this p-nitrophenol bound to basically glucose as our substrate. So go ahead and go to the next slide. In order to facilitate doing this from the ecology teaching point of view, Bio-Rad developed a bunch of these kind of more or less like what I call playing cards for mushrooms. And so when I run this in classrooms, what I'll do is I'll get basically my plain deck from the Bio-Rad website, and then I can go to a lot of grocery stores now and find a diversity of fungi for students to work with. And so what I'll do is I'll put these cards out. So it gives them the name, the common name, the Latin name, and then the habitat. And I'll give them the kind of, high view talk about mushrooms with different ecological niches will have different levels of this enzyme that'll, basically break down cellulose faster. What I'm having you guys do is look for the one that breaks it down the fastest. And so I'll basically lay my mushrooms on these cards and then have the students get to select which ones they think are going to have the strongest cellulobiase activity based upon the habitat that it grows in, because that tells them something about its ecological niche, something about its evolutionary history. So most grocery stores now, if you go into like a typical Safeway, you can at least find the button mushroom. You can often now find chanterelles, surprisingly, and oyster mushrooms. And in about 50% of them, I can also find shiitake. If you live in some of these major cities and you go to like a Whole Foods or more of a high-end retailer, you'll usually find a much bigger diversity. And so our slide deck basically has that full diversity in there, so you can just print those off and use those as your primer points for students to think about the ecology that their fungi that they're going to test come out of. Let's go ahead and go to the next one. So one of the big things, though, that I find that students need to understand for this is in order to get the enzyme out of the mushroom, you've really got to crush it up, because cellulobiase, like I said, not many organisms have it. And so mushrooms really exploit this to their advantage, which means they will digest things in their environment with enzymes, right? Because they don't have a stomach. So they literally just kind of dump out their stomachs, their enzymes, into the environment around them. But if they were to break it down all the way into glucose, they then would have to be fighting bacteria for that glucose. So they don't do that. What they'll do is they'll break cellulose around them all the way down to cellulobiose, then they'll import it. And inside their cells is where they'll break it down to glucose. And it even goes one step further, which is they will transport that cellulobiose all the way to the cells that actually need that energy before they start to break it down. So if you've got like your traditional mushroom, what they will do is they will basically move that cellulobiose all the way up through the stem, all the way to where the gills are and the cap of the mushroom is, because that's where that energy is needed to produce the spores and the sexual activity of the mushroom. And so even like in a mushroom like this, you would find different values of the enzyme activity from the stem versus kind of the cap material, which is something to be aware of. But what you really need to drive home with students is they really need to kind of get that mushroom out of there by grinding it up. And so for the protocol that Bio-Rad uses for this, we recommend basically a mortar and pestle system. And so what students will do is they'll collect their mushrooms, and you could use fresh mushrooms. You can also use dried mushrooms, which is something great to know with this lab, because if you want to do it where they get to go out and pick their own, you can decouple when you do the picking of mushrooms from when you run it. And all you need to do is basically just rehydrate your mushroom in a little bit of water the hour before, and now you get that fruiting body to work with again. So in the Bio-Rad protocol, what we have you do is basically get about a 2-gram piece of the mushroom, and they're going to grind that up with an extraction buffer that comes in the kit. And so they're basically going to grind it up with 2 milliliters of extraction buffer and just grind the heck out of it. I usually tell students I want you to grind for at least 1 to 2 minutes just to make sure it's all the way ground up. A trick though, and this is just because I really hate cleaning mortars and pestles, is if you are like me and you want to basically lower the amount of cleaning you do, you can also use miniature mortars and pestles, i.e. you could use a micro centrifuge tube with the extraction buffer in it. and you're just going to keep the same ratio, which is basically, 1 gram to 1 mil. And so I'll decrease this down to basically 0.2 grams of the mushroom now with an equal volume of my extraction buffer. And I can just use the bent micro pipette tip, and I can use this now as my crushing. So I can crush up in here, and it saves me from having to clean all those mortars and pestles at the end. However, though, since you're crushing all this material up and you're getting all these fibers everywhere, that is going to create a nightmare for a spectrophotometer. So if you're one of those teachers, which I am, I like to not tell my kids if they've had a chance to use a spectrophotometer before, and I'll cut them loose to do one small reaction with this. They'll put it in the spectrophotometer, and they'll be like, I'm getting these crazy values. I'm getting like 2s and 3s like that. And I'll just look at them and be like, whoa, what do you think's going on? And usually you can kind of coax them into it to looking at their sample and going like, oh, you know what? I see a bunch of fibers floating in there. Is that causing a problem? And then ask them, well, how could we solve that? Bio in their manual basically drives them right into what every biology teacher always does with anything that's cloudy, right? Put it through cheesecloth. But if you've got the time, allow the students to try and figure out how they could solution that. And they could do it a whole bunch of different ways. You could put it in a microcentrifuge tube and spin it. You could do the cheesecloth. I actually like to use just a tea bag. And so what I'll do is I'll basically just suck up my solution. I'll put it above a tube, and then I'll just squirt through the tea bag, and that's really quick. And I can collect the 250 microliters I need of the solution to move forward. But what you get to do there is allow students to do that troubleshooting, right? Which is really important in science, and you learn stuff. I always just, because I was bred and raised on cheesecloth, that's what I thought too. I had one kid one time take a card, and he would drip his material here and then collect it at the bottom. Did it work? Capture the fibers? Yeah. So give them that chance to kind of explore there if you've got the time for it. So once they've got their mushroom extract, let me show you what's at their lab stations just so that you know what they're working with. So all the lab stations are going to basically have a selection of reagents for them to work with. So we're going to have the substrate that we talked about at the beginning, that one that's going to give you that p-nitrophenol product at the end. They'll have a stop solution. All the stop solution is a slightly basic solution that will knock out the enzymatic reaction and allow p-nitrophenol to get you that yellow color. And then they'll have a tube to run the reaction with reaction in, and a control tube if they're running a control reaction. And then 2 basically transfer pipettes to do the pipetting with. They will also have a set of standards, and you can make up a class set and just do it once, or you can have one for every single lab station. And this is where that green screen becomes horrible, but basically it's p-nitrophenol going from 100 nanomoles down to 0, basically dropping by half each time. And they'll have a set of cuvettes where they're actually going to run their reactions in. And so what students will do is they will take the stop solution and they will preload all of their cuvettes with 500 microliters of the stop solution. And the reason that we do this is that way when their reaction's running, if they pull out the enzyme and that substrate being broken down, when they squirt it in here, it will basically kill the reaction and give them the color change. And so what we can do is develop a number of time points, basically from 1 minute to 8 minutes, where they will see that color change as product is being formed, as that cellulobiase is breaking down that substrate producing p-nitrophenol. One thing to think about though is if you're trying to get more of the eco teaching point from here, you might not do the whole time series. You might just go after in time points. So instead of doing 1 through 8 minutes, I might ask students, I want you just to do that in point at 8 minutes, but I'm going to give you a bigger diversity of fungi to play with and see how they all track against each other. It just depends on what learning objective you're trying to get out of that with the students. One thing to know is that after you run these reactions, they are very stable. And so you could basically cap these back up, put them in the box, and do the spectrophotometry the next day if your classroom time doesn't allow you to do the spectrum photometry work the day of. So those are things just to be aware of for that lab. And remember, this is just one activity of like 6 that are in the kit. So you have a lot of reagents to work with, which is great because if you really want to hone in on that bioprospecting portion, I mean, you've got pretty much like a huge supply of materials to work with. And you can even have them reach outside of fungi and say, where else do you see cellulose being broken down? Bring that in. Let's see if we can find activity there. So that in a really kind of quick nutshell is how the lab activity works. So you can go ahead and go on to the next one, John.
All right. So what I wanted to just remind everybody is that this lab that's found in that uses the Biofuel Enzyme Kit, you have to use the GoDirect Spectrobus Plus to look at it because the color is yellow. You can't use like our colorimeter, which is too bad. So that's why we tell you to use the GoDirect Spectrobus Plus. The Investigating Biology Through Inquiry book that has this in it is designed for AP biology, college biology, but it's done in an inquiry style. It has a ton of sample data in it and a ton of experiments. And I actually, in the folder that we've given you, luckily about 10 years ago, I saved all of that sample data. So I have put all of that sample data from Logger Pro into that folder, and then I've also converted it into graphical analysis so that you can take a look at it. But what I just wanted to show you next was just kind of how to collect data. So if I go to the next slide here, what I'm going to do, remember, I just wanted to say Vernier is for teachers, by teachers. Biorad has been wonderful to work with on this in this case. I'm going to show you how to use spectral analysis. This is going to be a live demo, just really quick to see what the data looks like. But then really what we're going to do is we're going to go to graphical analysis, and I'm going to show you how to insert a video. I've actually inserted a video into this data in Graphical Analysis Pro. And then I'm going to take some time and we can actually analyze together these data sets. So we can actually mimic together what you could actually do with your students if you were teaching remotely. So we're actually going to sort of practice that. And once again, just make sure you download GA 5.4. There's a link in the chat to get that, and we'll get to that there in a bit. So once again, what I'm going to show you, just to review, is I have samples of shiitake that I've processed. And remember, shiitake grows on dead oak trees. I also have some buttons that I've processed, Agaricus bisporus. It grows in fields or on manure. And then I also have some chanterelles that I've processed. Chanterelles are really cool because they are mycorrhizal, meaning they form associations with specific trees. I know that down in California, they tend to create mycorrhizal associations with oak trees. Up here, they're different ones. But you can imagine that maybe if you were a chanterelle and you've developed basically a symbiosis with an oak tree, you might not want to digest it. And so that's the really cool question that you can ask your students, is based on the 3 habitats that these things are found in, where they're found, what they live with, do you think they would process more cellulose or not? So right now what I'm going to do is I'm going to launch SA, and we're going to do a live demo, and I'm going to show you those 3 samples. So just be patient with me while I get that started. So let me launch SA. SA is what stands for spectral analysis. So this is spectral analysis. I don't know if any of you use IPEVO Visualizer, but I love it. Damon, let me know if you can't see my screen. I'm gonna switch. Great, thanks. I'm gonna switch to my GoPro webcam. There we go. And so here's my spectrometer. Let me put these side by side really quick. I'm just going to show, let's see. Oh, I have to see both. Okay, that's fine. I'll just put this right here. There we go. So I'm going to just start a new experiment. Once again, spectral analysis is free. So I have this spectrometer plugged into spectral analysis. I'm just going to select as a function of a full spectrum, but you can also do it in another method that I'll show you. So it's going to say, hey, the lamp needs to warm up for 20 seconds. The lamp's actually fine. It's been warmed up for a while. Now, this spectrometer can work on a LabQuest too. So if you have a LabQuest, you can do this experiment with the LabQuest. You can also do it with a with Logger Pro too. In either case, it doesn't matter. IPEVO Visualizer is just software that works super well with any camera. So I'm using that as an app to show you. Even though I've got a GoPro, I'm able to show these side by side. It's just a really, really good app. So it's IPEVO, actually, Visualizer. I can give you guys a link later. So I'm going to take the blank. Here's the blank. I'm going to put that in here. And I'm just going to calibrate my spectrometer. Okay, great. And then I'm going to take a look at, let's take a look at, this is a criminy or a button mushroom. Put that in the spectrometer. Let's collect a spectrum. There we go. Stop. We can take a look at it here, examine it. At about 405, it looks like it's got about 0.787. That's the absorbance. Let's take a look at Shiitake. So I'm just collecting these spectra on top of each other. Oh, look at that. Shiitake looks like it's bigger than the criminy mushroom. What about the chanterelle? What do you think? Any guesses? Anybody want to tell me what you think? Go ahead and put it in the chat there. Just go ahead and guess what you think. Be bigger, smaller. It's okay. You don't have to sit in the back of the room. You know, you can respond. It's really okay. Lower, yes, I like it. Let's see. Yes, much lower. Much lower. Right now, there's a way that you can actually set this up to show just an individual wavelength too. I don't want to spend too much time on that, but if I hit new experiment, I could say, oh, hey, let's just do this as a function of time. I could say, okay, I want to track an individual wavelength. I'm just going to put Shiitake in. This is a live readout down here of it, and I could click on it. And then like in the instructions, I could just pick like 403. Now you can see this live reading down here below. And what you could show your students is you could just pull one out, put another one in, and you're going to have a live readout down in the bottom corner. So there's different ways that you can use this. Now, the question that you might be asking yourself is, well, great, John, but how am I going to do this? You know, with COVID, I mean, I can show my students this, but how do I make this interactive? How do I make this happen so that if I'm teaching remotely or if I'm in class, they can see all of that? That's a really good question that we're going to answer next. So let me stop using SA. Let's go back to the live demo. So do you have any questions? Spectral Analysis is actually free. It doesn't cost anything. It's not like Logger Pro. You can get it free on our website. If you have one of our spectrometers, you can use it. It's super simple to use. What I wanted to just talk about next is we're going to explore what we call some remote solutions. So we have a whole remote solution on our website with lots of different data. Graphical Analysis, like I said, is free. If you haven't downloaded it, download it now. But I'm going to show you Graphical Analysis Pro. Now, Graphical Analysis Pro costs a little bit more, but it's an annual subscription that is based on the number of students that you have, but it can do some really amazing things. So what I'm going to try to go through is show you these three different, what we consider our learning environments, right? So here's in-person learning, right? Where you're the educator, you're presenting. That's kind of what I just demonstrated, right? Where every student's got a spectrometer, they're collecting data, you all do the experiment together. What about if you're not in that situation though? What if you're doing it asynchronous? Meaning students have to do the lab some other time away from you when you're not there teaching. Well, you can video sync data, or you can have video actually in GA Pro. So let me show you that. So I'm just going to show you that videos can be inserted into Graphical Analysis. And by the way, all of these files that I'm going to show you, they live in that folder. So we have given you access to these. So if you get GA Pro, you can use them. More importantly, though, even the sample data that I'm going to show you later, you could use without GA Pro. You would just, when we show you how to share it, that might be a little bit different, but we're going to be giving you lots of resources to use, hopefully. So let me launch GA. Let's close this. So here's Graphical Analysis. Now this is Graphical Analysis Pro. If you look up here, there's going to be a little radio button, and that's how you can tell it's GA Pro. If there's a little shield, it's normal GA. So I've created a file. I'm going to go here and I'm going to open Mushroom Video. So here what I have is I have a video of collecting spectra with those same 3 samples. I go through how to calibrate the device. Then I go through how to collect a spectrum. And then I go through how to set it up so the student could see the entire experiment. And then over here, I have the 3 samples. Now I haven't labeled them because GA can't deal with text, but you can actually guess which one is which. And you could even tell the student, hey, well, which one is this one over here? Well, that's got to be the Shiitake. Hey, which one is this one here? Well, that's got to be the crimini. And then which one is this one here? Hey, that's the chanterelle. And all of these numbers will correlate to the data that they're actually seeing. So this is a great resource. You can give this to your students. They can see the data, they can analyze it, etc. Do you have any questions on that? Like, it's just a really cool way to insert a video. You can sync data to video as well, but since this is a spectrometer, we can't do that. But that's another feature that you can do with other sensor data. Okay, what if you wanted to make this a little bit more complex, right? Like this seems like very basic. It's just I'm looking at relative levels. What if you wanted to do a little bit more math? Okay, well, we can do that. Let me open up a new experiment. Let's look at mushroom curve. So in this case, what I've done is this is the same video. So it's the same video that shows the crimini mushroom, the absorbance of crimini, chanterelle, and shiitake. But what I have here is I have the data from the standard curve from the Biorad kit. So for example, we could go ahead here, move up to the video here, and I could say, okay, let's find, let's see, where's a good sample here? There we go. There's the crimini mushroom. So you can have the students forward through the video and pause and find the absorbance down here for each mushroom. And then you could say, okay, I want you to go over here and I want you to analyze this standard curve. And they could go over here and say, okay, I'm going to apply a curve fit. There's a linear fit. And then if you were like me as a former instructor, I would say, great. Now that you know that the absorbance is 0.773, use the math to calculate how many micromoles of paranitrophenol were produced. And then after they had shown me that they could do that, I would say, great, now I'm going to make your life a little bit easier. Go over here, go to, where is it? Let's see, edit graph options. Oh no, there it is. Go select interpolate. And now you can just go and find that on the actual graph. You could go to 0.773. There we go, 773, right around there. And it's going to show you right down here, that's about 44.515 micromoles. So this is just a really interesting way for you to insert video. And if you're teaching asynchronously, you can actually create or have them do interesting data, look at it, and have them do more what I would call lab activities. So are there any questions? This is just how to insert video and how to analyze video. Now, this is super easy to do. If you have GA Pro, let's just say you did a new experiment, don't save. Oh, what was in the blank cuvette? What was in the blank cuvette is just the is really just the buffer And then I think it's a really, really small amount. You're basically using the buffer as your blank. That's all actually in the instructions as well as what the blank is, but you would want to use the buffer. Let's see here. So I could do new experiment, manual entry. And then if I click over here, I can turn on video, and then I could just import any video. So I could then go and import a video. Where do I have on my desktop here? Where is mushrooms? Oh, there's spec test. And so that's just that video that I was using. So you could also create your own videos. You could make them, I just did this all on my iPhone. So it's super simple and easy to do. So let me stop that and let's go back to the presentation. Okay, so what if you're actually, what if you want to actually teach with a class like in a Zoom and you're not asynchronous? So that's what this example is right here. That's a little trickier, right? I mean, that's not just giving a student a file, having them open it and having them take a look at it. This is a little bit more complicated. So I'm going to show you another example of that, which is, this is actually really unique. We've just started offering this. So what you can do in GA Pro is I can send data to you over the internet. And I'm going to demonstrate that here in a bit. So what you can do is you can actually create, you can open GA as the instructor. You can then create a session, you give the students a little code, and then you can send that data out to them. Sadly, the colorimeter will not work with this experiment because it doesn't have the proper wavelength. I wish it did. So once again, just download GA Pro, and we're going to go on to that next here. So what I'm going to do is I'm going to show you how to set all of this up. And what you probably are going to want to do is write down this number over here. This is 3 capital 0, lowercase l, lowercase I, capital EXN, DV. And you're going to need that later, and Angie has put that into the panel. And by the way, this is going to allow you to use Graphical Analysis free for an entire week, Graphical Analysis Pro. And I'm going to show you the teacher view, and then I'm going to show you the student view, hopefully. All right, so I'm going to go back to GA. Now what I'm going to do here is I'm going to get rid of my key. So I'll show you what you're actually going to do. So let's see, I'm going to get rid of my key, 10000. So you as the instructor would see this shield, right? And that when you open up GA right now, and what you can do is you can tap on the shield and you can insert this code, 3 capital 0, lowercase l, I, EX, N, DV. And there we go. Now I have access to GA Pro. There's a lot of cool things in GA Pro. There are like all these experiments, other things, like if you wanted to see live experiments, we actually have experiments linked to video. But what I want to do is show you how to share a session. And I want you to actually analyze this with me if we have time. So what I'm going to do is I'm going to choose a file to just open up. Where did my mushrooms go? I'm going to go to desktop. There we go, mushrooms. Let's just open up mushrooms evolution. There we go. So here I have the actual data sets that David and I were talking about. So these are those time-based data sets. So I've collected data as a function of time. And maybe you want to give these to your students, right? You want to give these and have them take a look at them. So I'm going to move this over here. What you can do is if you click on this, we call it a TIE fighter for Star Wars. But if you tap on this little icon up here and tap on data sharing, you can start a session. And then you can type in this little number. Now, if you can go ahead and do this right now if you want to. I'm going to show you how to do it. You can do this with me. I just need to go over to my Chromebook and I'm going to move it over into the screen so we can all see it together. So this might take me a little bit of time. So I'm going to go over here to my Chromebook, and I'm going to cast it. And then I need to go grab it. So I'll bring it over here. And hopefully you can all see it pretty well. Damon, can you see that okay? Great. So I'll flip between these. So the order code here is QU, this ID code is QUJ8 Northwest, okay, NW. So then as a student, what you would do is you would tap here. Actually, let me do that on the screen here. Oh, there we go. The student would launch GA, and they're going to say new experiment. And then they're going to select data sharing. And this is what I want you to do so you can pretend to be the student. You can actually open GA, select data sharing. Oh, wait, there we go. I have to put in the code again. Sorry there. I have to remember, you're going to have to do this too. Put in, there it is. There's the code, the GA license code to submit. It's 3, capital O, lowercase l, lowercase I, capital E, capital X, capital N, DV. And then if I submit, now I'm in GA Pro. Now I can do this. I'm going to say new experiment, data share, data sharing. And then I can enter the source code, which is over there. It's QUJ8NW. So let's see, QUJ8NW connect. Wow, see, that's the data is now on my Chromebook. And why don't you all try that right now? So you can do that and we can analyze the data together so you can see that it works. So go ahead and analyze it, take a look at it, and you can pretend to be the students, and then I can go back to the other session. Let me know if it works. I'm gonna wait right here for a few minutes and just let me know if it actually worked for you. So did it work? Did everybody get it to work? Any questions? So if I move this over, what you can actually do is analyze that data, just like I am right now. So I can turn on the graph legend, and you can see I have the red is the criminy mushroom or button, the shiitake is in blue, and the chanterelle is in yellow. And I can actually analyze those. I can apply a curve fit, and I could compare fits. Now, what I've done is in that folder, there is not only data for this, but there's not just this data, there's also a preliminary activity, which is just for the criminy mushroom. I've also put in data that includes data for the different parts of the mushroom. So the stalk and the gills and things like that. And then also in Logger Pro, there is like every single data set that I created when I created this lab activity. And if you wanted to create your own data sets, all you have to do is open up Logger Pro and copy and paste into GA, or to what we call graphical analysis. So do you have any questions? Was everybody sort of able to get that to work? Any questions? Feels awful quiet. All right, I'm just gonna assume that worked for you. Let's go back to the presentation. Now, there are all of these other remote solutions. So what we have at Vernier is we have all of these remote solutions for you. We put these up back in March. If you look at high school or college, we have all of these sample data sets. So there are tons of sample data sets that exist, and they are free. So here's GA Pro, you could get that, but we have lots and lots of experimental data, lots of biology experimental data. Once again, like all of the sample data that exists in our inquiry biology book, I haven't been able to put it in this sample data library, but if you're interested, just ask and I can get it for you if you're interested. There are other Darwin Days activities. So we have an entire web page that's devoted to Darwin Day and different activities and all sorts of different activities that you can do. So like this is a really cool little activity where you compare using a dissolved oxygen probe, a betta, which is a mouth-breathing little fish, a snail, and then a goldfish. And then we have a whole series of other activities. One of them even is another Bio-Rad activity, which uses their SDS page activity to look at molecular evolution of fish, which is really great. And then I also wanted to mention that you can use other Bio-Rad kits with this activity. One of the confounding variables, or one of the variables you might need to consider, is that how much protein do you have in a piece of chanterelle versus a piece of shiitake or something else? And you can actually use the GOT protein kit and the spectrometer to actually control for that, and then to see, well, is there really that much? If you control for the amount of protein, then you can actually control for the amount of enzyme that you have. And that data set is also in the data set that we gave you. Let's see here. And so with that, what I wanted to let you know is this is a free 30-day trial of GA. You can sign up for a free 30-day trial if you want. And then if you have any questions, please put them in the Q&A right now. You can always get in touch with us at all of these in all of these ways at Vernier.com. Here's the 1-800 number. You can email support@Vernier.com. We have chat. Even more importantly, though, is if you want to talk to a biologist, like maybe you want to get in touch with me or one of the biology team, you can just email biology@Vernier.com, and that will get to one of us directly right away. And so with that, we'll take questions. Any questions? Do you have anything you want to add, Damon?
I was going to actually point out that with the 3 mushrooms you selected, they're the quintessential ones if you're trying to show those different kind of niches. And the beauty about this kit and extracting from mushrooms is that you'll get variability depending upon who's doing the extraction every time, right? But those error bars, for the most part, aren't going to cross over on kind of these 3 species, just because you've got one that's, shiitake is the best mushroom you can find day in and day out as far as cellulose bias activity goes. And then chanterelles or boletes like porcinis will be that almost, nothing's registering. And then the button mushroom will be middle of the road. So if you're looking for the 3 stellar ones to grab, what Melville just showed you are the ones to go after. A great thing, which is that variability can be nice, because if you have multiple student groups, say, go after the crimini button mushroom, and they can see that variability, you can talk about now, how does that come to be through the way that protocols are developed and processed, right? So you can kind of dig into what causes variation within experiments too.
Yeah, what I wanted to, I'll make sure that I drop this into that folder to tell you about too, is I actually have a data set that I put in here that you could also use with your students. These are wild. I used to pick mushrooms when I was in college. So I've got Stropharia ambigua, Russell brevipies, a coral fungus, and then bluets. So you could also give this whole data set to your students and just say, well, go research, go find out where they live. Does this data make sense? So there's lots of different ways that you can make this creative and inquiry-based, even if you can't be with your students right there in the classroom.
Yep. And with a number of these too, if you want to also engage this at a citizen science level, you could have them interact with the iNaturalist platform to either document ones that are around their home, or in the case where John's got the data sets for bluets and russulas, things like that, they can actually go onto iNaturalist and find observations of those around where they live. And so they can actually see the habitat with the location that they live in, which is kind of cool.
And then once again, I'm just going to drop all of this data should already be in that Google folder. Angie gave you a link to that, but I will drop everything in there when we're done. update it, make sure everything's there together, and then you can just go in and grab all that. Angie, how much longer is that Google folder going to be available for everyone? I assume well until like next week. Is that correct?
Definitely, it'll be open for a lot longer. So don't feel like you have to rush to access the documents, that it will be available for a while.
What other organisms were you thinking of there, Damon?
So I've had some teachers actually use, cow pies per se, because there's cellulose being broken down there. I've had some do termites. I had somebody in a county over from me actually start multiple compost heaps at the start of the school year, and then they sampled those with this kit at the end of the year to see. We started with a bunch of like woody substrate versus straw. Did we see a big difference in who shows up over the course of the year? So there's some definitely, you can allow them to get creative with where, cellular bias might be present.
Yeah, the protocol might have, well, does the protocol differ?
Not really, because it's pretty much the same, right? Take organism, grind to then try extract the enzyme from it and then filter. And so that's the generalized protocol you can use with a lot of different organisms. And I have some teachers that like to just throw, a red herring in there, and they'll give them like celery and say, see if you can find it in here. So it is good to kind of throw a couple of those in there too, I think, just to kind of reality check that this thing's not all over the place and you're specifically looking at these organisms because they are the ones that really do produce it.
Yeah, that's the other thing I'll bring up, that the biofuels activity, the biofuels kit is actually used in 2 labs in that inquiry biology book. We have an entire just biofuels activity where you look at biofuels as well. So we try to make sure that in that book, there's more than just one activity that you can do. Now, how many people does that one kit support, Damon?
So the kit supports 8 workstations for 6 activities. But if you're just really interested in doing the eco portion of this, out of one kit, you would have enough liquid reagents to provide for at least 30 students. You would just have to get a couple more 15 mil conical tubes and things like that if you're sending out individual student stations. If you're going down that route, send me an email and I can help kind of tailor it out to the number of students and things like that. Because, hopefully we're not in this for that much longer, but while we all have to make adaptations, right?
And they can buy a refill kit too, right?
Correct. So you can buy just the reagent baggie, which is perfect. So most of my customers I work with in year out, they'll buy the kit the first time that has all the plastics and stuff. And then once they've kind of figured out what they need, they're just down to just buying the reagent baggie every year. The beauty about this kit that I really like is that from the prep point of view, is that these reagents are really stable. So even that substrate, and this is my biggest worry with most enzymatic reactions, is the substrate I prep isn't going to hang around for very long. I've prepped this and held on to it for weeks at a time just in the fridge and haven't had any problems with it breaking down. So it's a really robust system from what happens in real life with stuff, right?
And to get to Damon's comment, these samples that I have, I made them 8 years ago. So like I seal them and I take them with me on shows. So that's another thing as an instructor, you could just, this is just like the little stuff that you put on the end of, it's just a little rubberized stuff that they sell for putting on tools and stuff. You can make samples and save them as well to use for demonstrations, but it's really, really stable. It's a nice little lab to do for sure. Is there any special disposal of reagents that they need to worry about?
There's nothing you have to worry about with this one, which is great. Bio-Rad goes to the ends of the earth to make sure things are relatively easy to dispose of most of our kits.
Yeah, the only thing that I would say once again is that if you have time to go to a real, once this is all over, if you have time to see this done in quote unquote real life, it's great. The key thing to remember is, as Damon said, you're not going to get the reaction to change unless you add the base. So that's one of the things that can be a little bit tricky is you're doing the reaction, then you add something to stop it. So we typically call this a stop reaction. The base stops the protein and enzyme at the same time and then turns it a color. So that's really the only trick that I found when I've done it with groups is you have to make sure the students are used to, if you're going to do a time series, stopping it every minute. that's why I think Damon's idea of actually doing it where you just do the end reaction is just beautiful, because you can say, nope, I want all of you to wait for 5 minutes or 6 minutes or 8 minutes and then stop all the reactions, and you can look at all sorts of different mushrooms. It's a great way to do that activity. So if that's it, I think we're like pretty close to time.
Hey, John, there was just one question about the CMBL files. If you can talk about how to access those, they weren't able to open it.
Okay, great. So what I've done, since I couldn't copy everything, the CMBL files are Logger Pro files. Now there's a free 30-day demo of Logger Pro on the website. It doesn't cost anything to get access to it. And what you can do is you can download Logger Pro onto your computer or your Mac. You'll be able to see that data, and then there's columns of data that you can just copy and paste into GA. I was going to show you that you can data share from Logger Pro into GA, but sometimes that gets a little confusing. But all that I wanted to do is make sure that you had access to all of the data that I had from 10 years ago. So you can actually open that up and you can just copy, and you can even copy and paste that data into a Google spreadsheet. I just wanted to make sure that you have everything, because I wasn't able to transfer everything over into GA so that you can see it. So I just wanted you to have access to it. So just download, go to our website, vernier.com, search for Logger Pro demo. If you don't have Logger Pro, download the demo, and then you'll be able to open up those .cmbl files. .gambl is for GA Pro, and then non-GA is as AMBL files. And if you have any questions, like let's say you can't get into them or you have a question, remember, just email me at biology@vernier.com. So that's just biology@vernier.com, and just say, hey, John, how do I get access to that? I'll send you links, get you whatever you need. All right, anything else?
I know we're a few minutes over, so I think we should be mindful of people's time. So thank you, everybody, for joining us. Feel free to reach out to Damon or John. They put their contact information in the chat if you have any follow-up questions. So have a great weekend, everyone.
Thank you.
Take care, everybody.